I will start my class today. Okay. So what I will do first is share my screen. Post-translational transcriptional modifications. This is something which is happening, a type of gene regulation which is happening after your gene has been transcribed. So previously we have seen that during gene regulation we are just concentrating mostly on what happens during transcription initiation. At that time, how much is being expressed, although that is a major part, but still there are other factors which are also playing a role in gene regulation. So let us talk about certain transcriptional modification, post transcriptional modification. So almost all major types of RNA synthesized by cellular DNA depend upon RNA polymerase. And these undergo changes before they can carry out their functions. So any RNA by a DNA dependent RNA polymerase, there is something splicing, capping, tailing, methylation, some sort of modifications, they always take place before uh, uh, some transcription or activities take place later, before the particular phenotype is made. So all of these they have got these post transcriptional modification and processing. So what does it involve? It involves addition to or alteration of existing bases or sugars. Some bases are changed, some are added, some are removed. Some chemical changes are made, alterations are made to existing bases. These are all which happens after the transcription has taken place. The pre-nascent or heteronuclear RNA is already being produced. Next is the post-transcriptional processing. This involves phosphodiester bond cleavage and loss of certain nucleotides from this transcript. So what happens in post-transcriptional splicing? That is phosphodiester bond, there is a cleavage and certain regions are removed from this nascent RNA or heteronuclear RNA. The nascent RNA, also it is known as the primary transcript. It needs to be modified to become functional tRNAs, rRNAs and messenger RNAs. And this is a very critical system in eukaryotic system. This modification is something which defines the eukaryotic system. So what are the changes associated with primary transcript? Let us go through them. mRNA in prokaryotes, there is no precursor. A poly A tail is added, process in some cases specific cleavage by endonucleases, and after that the product is messenger RNA. In eukaryotes, an heteronuclear RNA is formed. Heteronuclear RNA is formed. Two to 14 kb long, longer than a RNA precursor. So before transcription starts to take place, of 2 to 14 long in mammals, 8 to 10 kb, 4 times longer than messenger RNA, that pre-nascent transcript is formed. It is at least 4 to 5 times longer than the final messenger RNA. And what are the modifications which take place? 5' capping methylation and polyadenylation. And splicing of introns takes place. This is for messenger RNA. In ribosomal RNA, in eukaryotes, pre-RNAs, the methylation takes place. Specific cleavage and splicing of introns take place. In prokaryotes, there is no. In prokaryotes, there is no. No. A second. Splicing taking place. Eukaryotes, you have this cleavage and splicing, but in prokaryotes it is not there. In transfer RNA, there are different other modifications which take place. So, what are the modifications of eukaryotic messenger RNA? Achha. Modifications of the heteronuclear RNA which are taking place. So, primary transcripts of messenger RNA are known as heteronucleus RNA. They are larger than matured messenger RNA by 4 to 5 folds, and modifications capping at the 5' end, tailing at the 3' end, mRNA splicing, RNA editing. So let us first look at 5' end modification. The 5' includes capping and methylation. So the type of methylated caps are, there are two types of methylated caps are found. First is the monomethylated caps. They are commonly found in most eukaryotic messenger RNA. Then you have trimethylated cap. It is less frequently observed only in some non-coding RNA, but it is highly conserved throughout the eukaryotes. So basically in the small snRNAs, small nuclear RNAs and telomeres, you can find this trimethylated cap, but especially for your mRNAs, you be bothered with monomethylated caps. So let us look at the monomethylated cap. So A. Shatkin and his co-workers in 1975, they found out that the 5' terminus in most viral cellular messenger RNA, they had this 5' CH3 cap. So the entire methylated oligonucleotide, it is known as cap structure. It happens in the nucleus. Of most mRNAs, monomethylated. There is only 7-methyl guanosine cap. Guanylyl group is added. So it is at the methylated at the 7th position. So in the trimethylated ones, additional methylation occurs at 2' position and at the 6th amino of the first adenine. So be bothered with monomethylated cap. So this is the sugar, this is the base, and this is the phosphate. So the enzyme guanylyl transferase, what it does, it attaches to the phosphate a 5' CH2 and a guanine caps. So you have a 7-methyl guanosine cap formed. So you need to practice this figure very well. So if you look at the structure of the eukaryotic cap, the full cap structure is present over here. So the DNA, the messenger, the RNA is over here, and at the 5' end, 5' phosphate, the 3rd phosphate, and this cap is attached. So basically this phosphate will not self-destruct this particular sequence. So it's like protecting the RNA. Otherwise RNA undergo autocatalytic activities. So this is stopped because you have some methyl inert nerd of a cap blocking the 5' end. So let us go to the next place which we must study. So what is the function of the 5' cap structure? The cap may function in the transport of messenger RNA from nucleus to cytoplasm. It protects mRNA from phosphatase and nuclease attack, and also from itself, and it is required for the productive elongation of transcription by RNase polymerase II. It helps in ribosomal attachment. So there are so many functions of this particular 5' cap. And the leader sequence and the cap binding protein CBP, it may play a role in enhancing and to stabilize the interaction of messenger RNA with the ribosome And of course in translation initiation, not transcription initiation, translation initiation process, ribosome binds on this particular messenger RNA, and this cap actually directs them that okay, go in that direction or go in this direction. So it is very, and also it holds the two subunits of the ribosomes in a much stronger manner. We did not study trimethylated cap. Next is the 3' polyadenylation. You can see that once it reaches the end, a hexanucleotide sequence AAU, it is necessary for cleavage to generate a 3' end for polyadenylation. So this is cut and poly A tail is added. Poly A tail, from the back it protects your backside. The cap protects your head and the poly A tail protects your backside. These are all the same things which are happening. So these are the poly A recognition site in yeast. You can see in an yeast RNA, you can find all these regions. And this is in mammals. This is the polyadenylation machinery. So can you see this structure here? This is 7-methyl guanosine cap. दिख रहा है structure? Hello?
Yes, sir.
Okay.
Yes, sir. Yes, sir. Yes, sir.
7-methyl guanosine. This is the transcript AUG, or this is where the transcript, the open reading frame starts. The protein is coding from here, this region. Then it ends with the poly A tail and it goes up to AAA. And this is 40S unit, EIF3. This is the translational unit which is formed over here. So the initiation factor EIF4G, it serves as a multi-purpose adapter. So what is this EIF4G? It attaches the tail to the head. This is an adapter. Small questions come from this. So দেখো এই ট্রান্সলেশন হওয়ার টাইমে এই রকম জিনিসটা তৈরি হয় সব পাকিয়ে. These are all ribosomes are translating. Many ribosomes are binding. It's not one ribosome, one protein. Many are making joining. They are synthesizing the protein, leaving and going. Another is joining. So there is like a multi-ribosome complex, polyribosomal complex. So this will actually end my talk today. I will again take the next class maybe this weekend if I have a time. I will tell you people. Do you people have any questions? Ache question? Hello? Hello? শুনতে পাচ্ছিস?
हाँ सर.
Okay, okay. Any
Yes, sir.
Okay, okay.
सर इसमें शॉर्ट क्वेश्चंस आएगा सर
हाँ
इस
सब दे
टाइप का या
दूंगा,
फिर
चिंता
पूरा
मत कर.
प्रोसेस
क्या
एक्सप्लेन
क्वेश्चन
करना
आएगा सब
पड़ेगा?
दे दूंगा. एकदम क्वेश्चन पेपर में जो
वो
क्वेश्चन
नहीं
आएगा
बोल
उसको
रहे.
छोड़ के बाकी सब क्वेश्चंस दे दूंगा.
नहीं सर वो नहीं बोल रहे हैं. जैसे एक होता है ना ओपन क्वेश्चंस की
हाँ
पूरा
हाँ
प्रोसेस आपको
डेफिनेटली
एक्सप्लेन
क्वेश्चन बैंक
करना
दे
है,
दूंगा तुम लोग को.
और एक होता है कि एकदम शॉर्ट
आई
क्वेश्चंस में वन
अंडरस्टैंड,
वर्ड
बस
आंसर
मुझे समझ
मिलेगा.
में आया, मजाक कर रहा हूँ. हाँ, आई विल गिव द होल थिंग टू यू. जितना टाइप्स ऑफ क्वेश्चंस विथ आंसर्स भी दे दूंगा उसका.
जी सर.
ठीक है. ओके, बाय बाय देन.
ओके सर. ओके सर. थैंक